There are three characteristics of every experiment that are very important to make sure that we have. The first one is randomization. All of the experimental units or subjects need to be randomly assigned to the different treatment groups available in any experiment. So in this experiment here, we're going to assume that we have two treatments. There's treatment 1 and treatment 2. And the idea of randomization is to make two groups that are essentially equal. By randomly picking who goes in each group, we're going to say that even though there are differences between the people in the different groups, that those differences are going to balance out. So over here we have 9 people, 9 subjects for the experiment. We need to figure out how we're going to get these people into the two different treatment groups. We could arbitrarily say, okay, these 4 we're going to throw in this one, and then these 5 we're going to throw in the other. The problem with that is, as we can see from the different types of little stick figures here, we have some sword people, some muscle people, some sad people, and some happy people. And it would be best if these differences kind of averaged out between the two groups. We'll talk about specific ways that we can try to make sure that we get kind of one of each in each group, or things like that later. But for now, the simplest form of an experiment uses purely random sorting to get people in the two different groups. So to use pure randomness, first thing we're going to want to do is number our 9 subjects. So we throw a little number over their head here, and if you had a list of names, you could do the same thing. You would just number each participant in the same way that if you were trying to select an SRS, you might number them so that you could use a random number generator. And then we take out our calculator. Obviously there are other ways to generate random numbers, but since this is a pretty good one, we'll use it. If we go to math, we go to the PRB area, and we go down to randint, the 5th one down. Then we just need to type in 1, because that's our first number we want to generate. Our lowest number is 1, comma 9, because 9 is the highest number we want to generate. So we want to randomly pick numbers between 1 and 9. And before I even start clicking enter, we'll notice that we have an odd number of people, yet we have only 2 treatment groups. So I'm going to arbitrarily say that treatment group 1 is going to get 5 people, treatment group 2 is going to get 4 people. And that's okay. Usually you want your 2 treatment groups to be roughly even, but they do not have to absolutely be the same size. That's one thing that people get caught up on sometimes, is that the 2 treatment groups do not need to be of identical size in order for you to still analyze the results. So let's say 5 in treatment group 1, 4 in treatment group 2. The first 5 numbers that we generate, we're going to throw in treatment group 1, the rest will be in 2. So let's go ahead and do this. One shortcut is if we actually add an extra number here, it'll generate that many random numbers for us. So we go like that, 6, 3, 9, and then we start getting some repeats. So let's go ahead and throw 6, 3, and 9 in there. And since we have repeats, we have to keep on going. So let's go ahead and hit enter, run it again. 7 is the next number we generated. And then we have 9 again, 6 again, 7 again, okay, 8. So now we have our 5 unique values here. And then that means the remaining 4 would end up going in the other study treatment, or the other group treatment 2. So we bring them over here. So that's how you would use randomization to split up the 2 groups. Now you can see this time it seemed like all of our sword people ended up in the same group. All of our muscle people ended up in the same group. That's what one of the problems is with a small sample size, is that kind of thing can happen. Ideally, these people will end up more spread out, and when you have a larger sample, they almost always will, because of just pure probability. But when you have a small sample, sometimes you run into that using pure randomization. One of the things that's interesting about randomization is that volunteers are okay most of the time. Now, when we think about an SRS or a typical survey, we think of voluntary surveys as a horrible, horrible thing, and they are. Voluntary surveys are bad. When you're doing an experiment, however, you usually don't have the ability to just pick random people from the population and force them to do some sort of experiment. So even if the volunteers that you end up getting for your experiment don't represent everyone, they're going to be evenly split in the two different treatment groups so that the two groups are effectively equal. The only thing different between those groups is going to be the one explanatory variable that you're testing. So looking back at our groups here, the claim we're trying to make is that this group of people right here and this group of people right here are effectively equal. If we were to run the same treatment on both of them, their results should be roughly the same. Now, in reality, of course, the two groups are never going to be exactly equal. You can see that these people look different than these people. They're not exactly the same. However, the process of randomly assigning people to these two groups says that so many times out of 100, let's say when you start doing confidence intervals on hypothesis tests with this type of thing, 95 times out of 100, these two groups are going to be effectively the same. So that's the idea, that's the goal. And now what we're saying with the volunteers is that even though the volunteers don't represent everyone, as long as you're randomly assigning them to the two treatments, these two groups are going to be roughly the same. They're going to be considered essentially equal for our purposes. And that means that if they're essentially equal, the only difference between them is the explanatory variable that we're testing. The explanatory variable being the different treatments. So this group is equal to this group, but then we apply treatment 1 over here and a treatment 2 over here, and now they're different. That's the only thing that makes them different, and that's why when we do an experiment, we're only comparing treatment versus treatment, because everything else we're saying is essentially the same. That's the key to randomization. The time where volunteers are not so great. If you take, here's an example, if you take volunteers for women's hair products and mostly men volunteer, the results might be valid. You might have a valid experiment, except that the results only apply to men. So if you're asking for volunteers to do some sort of study, you have to make sure that your volunteers come from the population that you're interested in. Another example is that a lot of medical studies are done on rats. Rats are not humans. The reason we use rats is because if we try some kind of medical experiment and something goes terribly wrong and all the rats die, it's somewhat sad, but not really a huge catastrophe. If we have a bunch of humans in some medical study and something goes terribly wrong, that would be awful. And the ethical complications around that would be very, very severe. So that's why, even though rats are not humans, the stage 1 would be to say, alright, let's make sure it works in rats. Let's make sure it works in maybe larger animals, primates perhaps, and you kind of work your way up the chain until you're working safely in humans. So if you can prove that it works safely in rats, now we'll try a different animal. Then eventually we'll get to humans and make sure that it works in humans too. The second major thing, so first one was randomization. The second major thing is the idea of a control. A control is a group of people that you compare your results to. So in order to be an experiment, you have to compare the results of your new treatment to something else. So I develop a new pill, or I develop a new surgery, whatever it might be. I cannot just measure the effects of what happens to the people that used my new surgery, used my new pill. I need to compare it to something else. I have to be comparing. So the group using my new treatment as compared to a group using nothing, a group using a competitor's project, a group using an earlier version, all of these would be considered acceptable. Now the nothing group is probably an interesting thing to compare to. If you're comparing to a group doing nothing, you would say that doesn't really make sense. Later on we'll talk about, I guess, a more sophisticated version of the nothing group called a placebo, where people think they are taking a pill, but in fact they're just taking something that doesn't do anything medically. So having a group to compare to that looks like they're doing something but in fact medically is not would be one way, or thinks that they're part of the experiment, thinks they're doing the active part of the experiment but really are not, is a way to trick them into thinking they're participating, but in fact you're just using that group to compare. So let's go ahead and define control group right away. A treatment group where the experimental units or subjects have nothing new done to them. That's the key to a control group. It's a treatment group, just like the active treatment, the new whatever you've developed, but nothing new is done to them. They are purely there to serve as a comparison. And I think when we start getting to more examples of experiments, we'll see how this control group can have an important effect. You need to have something to compare to in every single study. Last thing is repetition, repetition, repetition. If you aren't able to repeat the results of your experiments, then it's not going to be very meaningful. There's lots of different levels to repetition. The simplest one is that you need to have a large enough sample size in your study. When we go back to this first slide here, how we ended up randomly sorting all the sword people into one group and all the muscle people into a totally separate treatment group, these two groups are not exactly equal. If your sample size is too small, that can happen quite a bit. And so if you don't have a large enough sample size, then even random assignment, as we used earlier, cannot stop you from confounding other variables, from having other lurking variables causing differences between your two groups. So you need to have a large enough sample size. Other things that come up would be repeating your study in multiple locations, repeating your study with multiple different people running it, various things like that, so that you can verify that in fact it wasn't just this one place or this one day or this one group of people that was special in some way, but that your results apply everywhere. So repetition is one of the big things that comes up to prove any scientific results. Let's look at this long example story and try to find the randomization, control, and repetition and how they're used in it. A grocery store wants to determine if they would get people to buy more oranges if they placed an extra stand of oranges near the dairy aisle. So they want to see if people will buy more oranges. Underline right there, that's what they want to measure. And their way to figure that out is having an extra stand of oranges near the dairy aisle. To run their test, they randomly select 3 days in a week to have oranges in only their standard place, so that's one of the ways they compare it to, and the other days they have the extra stand. On the standard days, data was collected from a whole bunch of people, over 2,000 people, that walked through the store. And on the days of the new stand, data was collected from over 2,000 people who walked through. Let's start with the middle one down here, control. There is some sort of comparison being done. The main thing that we're trying to measure here is how many oranges people are going to buy, and we want to know if the extra stand near the dairy aisle is helping. The control is something to compare this extra stand to, and it says 3 days a week they're going to have only their standard place where they sell oranges. So they're going to pick 3 days in a week and compare those 3 days from the standard place to the days where they have the extra stand. So the standard place, let's go ahead and add that in here, would be our control. The randomization is a matter of selecting who is going to get the control group, the standard place, and who is going to get the new experimental group, the extra stand. It says, let let's see if we can figure out how they sort those out. To run the test, they randomly select 3 days in a week. Let's highlight that. They randomly select 3 days in a week. So out of the 7 days in the week, 3 of the days are randomly picked. And that's how they break it down on that side. And another form of randomization that isn't maybe quite as apparent is the idea that people could randomly show up at the grocery store on any given day, possibly multiple days. That's not a very strict form of randomization, and in fact probably not a great measure, but it does have at least a hint of randomization going on there. The key one is, though, that they are selecting 3 random days in the week to have the oranges in only the standard place, the other 4 days randomly being selected to have the new stand. Last one is repetition. Couple ways repetition are used here. First one is the days of the week. The days of the week, it says there are 3 random days here, and if it's a week, then there'd be 4 random days for the other group. So the number of different days it's being repeated, instead of only using 1 repeat. Instead of only testing the new stand on 1 day and the old setup on 1 day, they have multiple days where they're repeating this experiment. The other way is the sample size. We have, on any given day, we have a lot of people. It looks like over 500 people walking through the store. So that's a lot of data. Let's look at one last example here. We have a teacher who wants to understand the effects of project-centered learning. And let's work strictly with this description. To do so, he observes the test scores of 22 students in class during the most recent unit. Control, it doesn't say anything about comparing these students to anyone else. Just observing the test scores of these students. So there's no comparison, there's no control. No past data to look at, anything like that. So the control is an issue. If there's no comparison group, there is no randomization, because you have to randomly assign people to different control groups. So if there is no control, it's going to be impossible to randomly put people into the 2 different groups. So no control, no randomization. Repetition, well, there are 22 students in class, but it looks like this study is only being done on the most recent unit, so at 1 unit. So the repetition is not particularly good. The sample size is okay. 22 is not a terrible sample size for an experiment, but not having any other units to compare to or anything like that makes the repetition pretty bad. So if there was a study like this, if there was an experiment like this, this would be a very, very poor experiment.